In the process of transcription, a section of DNA known as the gene is read and copied into RNA. DNA has two strands, the template strand and the non-template strand. Both strands contain genetic sequences, but RNA polymerase, the enzyme responsible for transcription, can only read DNA in one direction, from 3' to 5'. Let's take a look at gene 1. Here, RNA polymerase moves along the template strand from the 3' end to the 5' end. As it moves, it synthesizes RNA in the 5' to 3' direction, which is complementary to the template strand sequence. The process starts at a region called the promoter, moves through the structural gene, and ends at the terminator. The RNA produced is a complementary copy of the template strand. Now let's look at gene 2. The key thing to remember is that RNA polymerase always reads the DNA template strand in the 3' to 5' direction, no matter which strand of DNA the gene is on. In this case, RNA polymerase is moving in the opposite direction compared to gene 1, but it's still reading the template strand from 3' to 5', producing an RNA strand complementary to this new template strand. The important takeaway here is that while both DNA strands contain genetic sequences, RNA polymerase can only read in one direction, 3' to 5'. It can read different strands of DNA, but its direction of movement along the template strand is always the same. Notice that in gene 2, the positions of the promoter structural gene and terminator are reversed compared to gene 1. This is because the gene is on the opposite strand. However, RNA polymerase continues to move from 3' to 5' along the template strand. In summary, both strands of DNA hold important genetic information, but RNA polymerase always reads in the 3' to 5' direction, no matter which strand is used as the template. This directional reading ensures that RNA is properly synthesized, which is essential for gene expression.